Welcome to the deep dive. Today, we're exploring a really fascinating kind of hidden world, the world of what people often call slime molds. Specifically, though, we're zeroing in on one group, the Dictyosteliomycota, or the Dictyosteliid cellular slime molds. Our source material is a chapter from an Introductory Mycology text, which is interesting because, well, as we'll find out, they're not exactly what that name might suggest. Okay, so let's unpack this. What is it that makes these organisms, these Dictyostelids, so special?
Well, the first thing, like you hinted, is despite that common name, slime mold, they aren't really fungi. Not in the way biologists classify things. They're actually their own distinct group. Super interesting organisms that are saprobic.
Saprobic, meaning they eat dead stuff.
Exactly. They live on decaying organic material. Think least litter, soil, even animal dung. Basically, anywhere you find organic debris, and this is key, bacteria. Bacteria are their main food. And they're often missed just because they're so tiny. I mean, their basic form is just a simple single amoeba. But the really amazing part is how these simple cells get together and show these incredibly complex social behaviors.
Okay, so let's zoom in on that basic unit. What are these individual amoeba like structurally?
Right, so their main feeding and growing stage, what we call the somatic phase, it's entirely microscopic. We're talking about a tiny single cell. It's got one nucleus, it's naked, meaning no rigid wall, and it's haploid.
Haploid, like sperm or egg cells.
Precisely, just one set of chromosomes, and it looks, like a little blob. It moves using these temporary extensions called pseudopodia and just engulfs bacteria to eat.
And you mentioned they have these fructifications or sorocarps, the spore things. The text says they're usually tiny, easy to miss. But it also mentions some can be almost microscopic heaps, while others, like Polysphondylium, can form these delicate branched structures, maybe even a centimeter tall.
It's right. So while the active feeding amoeba is always microscopic, the structure they build to reproduce, the sorocarp, can sometimes be visible, but yeah, even then, they're pretty small. That slime mold name can be confusing, conjuring images of much bigger things, but dictyostelids are generally quite inconspicuous.
Okay, here's where it gets, for me anyway, really interesting. These individual amoebae, they come together to form this pseudoplasmodium, or a grex, or a slug, the book calls it. How do these separate little cells know to do that? What's the trigger? What's the signal?
That is the million-dollar question. And it's why they became such important model organisms, especially Dictyostelium discoideum.
Mm-hmm.
So what happens is when the bacteria they eat start to run out, the amoebae stop feeding, and then some of them just spontaneously become aggregation centers.
Mm-hmm.
They start pumping out a chemical signal.
The chemical call sign.
Sort of, yeah.
Yeah.
And the other amoebae sense this chemical gradient in their environment and start moving towards the source, forming these distinct streams. It's incredibly coordinated.
And this signal has a name. Acrocin, is that right? That's kind of mythical.
It does.
Yes.
Acrocin.
Yeah.
And for Dictyostelium and discoideum and a lot of its relatives, we know exactly what it is. Cyclic AMP, or can't amber.
CAMP, that's involved in signaling in loads of organisms, even us.
Exactly. It's a fundamental molecule. And it's not just a constant signal. The center pulses it out, and the amoebae moving in relay the signal, amplify it, creating these waves that draw everyone in. It's like a chemical chain reaction.
Wow.
Yeah. And while CAMP is common for Dictyostelium, other Dictyostelid species actually use different chemicals as their specific acrocin.
That level of cooperation is just mind-boggling for single cells. So, okay, they form this slug, it moves, but then the source mentions cellular differentiation. Things changed inside the slug.
This is maybe the most remarkable part. Before they even build the final fruiting body, the cells inside that slug start to specialize. It's like they commit to different jobs. Roughly, the front third become pre-stalk cells. They're destined, literally, to form the stalk.
Okay.
And the back two-thirds or so, those are the prespore cells. They're going to become the spores that get dispersed.
And are they actually different at that stage, like chemically or metabolically?
Oh, absolutely. The pre-stalk cells are buzzing with activity. They start making cellulose, which is needed for the stalk structure, and they produce ammonia. But here's the incredible part. These pre-stalk cells essentially sacrifice themselves. They undergo programmed cell death to become the rigid stalk.
Wow, really? They die for the others.
Yeah. It's an amazing example of cellular altruism,
Yeah.
Some cells give up everything so that the prespore cells have a better chance of survival and dispersal. It gives us clues about how multicellular cooperation might have evolved. The prespore cells, meanwhile, they load up on nitrogen, develop these special prespore vacuoles to make the tough spore wall materials, and basically prepare for dormancy.
So the slug finds a spot, the cells know their jobs, How does it actually transform into that stalk and spore structure, this culmination part?
Right, culmination. So the slug stops moving, rounds up a bit, flattens at the base. Then imagine this. The prestalk cells, which are now at the top, start diving down through the mass of prespore cells. They form a tube of cellulose as they go down. As this stalk tube gets longer, pushing downwards, it effectively lifts the mass of prespore cells upwards.
Like a little elevator made of dying cells.
Exactly like that. A biological elevator. Once the prespore cells reach the top, they mature into spores ready to be released. And the whole process, this intricate dance, is orchestrated by signals like KMP again, and another one called DIF, differentiation-inducing factor.
That's incredible. Okay, stepping back from the social drama, how do they just normally grow and multiply? The simple way.
The basic asexual cycle is pretty straightforward. A spore lands somewhere suitable, it germinates. A single haploid amoeba pops out, starts eating bacteria, grows bigger, and then divides into two identical amoebae through mitosis and cytokinesis. Standard cell division, repeat.
Simple enough. But the text also mentions microcysts. What are those about? Another way to reproduce?
Not quite reproduction, more like a survival pod. If conditions get tough, maybe too dry, too much ammonia built up, an individual amoeba can just encyst.
Yeah.
Rounds up, secretes a thin cellulose wall around itself, and becomes a dormant microcyst. It just waits it out.
Smart.
Yeah, very. When things get better, it germinates, the amoeba comes out, and carries on as before.
Okay. Asexual covered, survival covered. What about sex? The source brings up macrocyst formation, but also says people used to doubt it even happened.
That's true. For a while, especially in some species, scientists weren't sure if they really underwent sexual reproduction. But yeah, it's now confirmed for several, including Dictyostelium. It starts when two compatible amoebae fuse. They form a diploid giant cell, basically a zygote.
Okay. Typical start for sexual reproduction.
Right, but then it gets weird. This giant cell acts like a magnet. It attracts lots of other normal amoebae towards it, and then it engulfs them. It pulls them inside itself.
It eats them.
It does. It surrounds itself and all these captured amoebae with a wall, a primary wall. And inside this walled structure, the giant cell basically has a feast, consuming all the amoebae it trapped. It's pretty wild sort of cellular cannibalism within a cyst.
That is unexpected.
Definitely. After it's done feeding, it lays down more wall layers, forming the mature macrocyst. Inside, the zygote nucleus undergoes meiosis, then mitosis, producing lots of new haploid amoebae.
Yeah.
After some time, could be weeks, this macrocyst breaks open, releasing all these new amoebae to start the cycle again.
So it is sexual reproduction, just with a dramatic feeding phase.
Pretty much. Though the text notes these macrocysts can be tricky to get to germinate in the lab, so their exact importance in nature is still a bit debated. But we know they exist, both self-fertilizing types and types that need a partner.
Okay, let's switch gears a bit. Where do these things actually live? We said decaying matter.
Yep. Very common in dung, soil, especially forest soil, decaying logs, old mushrooms, leaf litter, anywhere bacteria and decomposing plant stuff are plentiful.
And are they found everywhere?
Remarkably so. They have a huge global distribution. Temperate forests, sure, but also deserts, grasslands. People have even found them in Alaskan tundra. The mix of species you find definitely changes depending on the habitat, though. Makes sense ecologically.
How do they get around? They're microscopic.
Right. The spores are often held in this sticky droplet, which is perfect for hitching a ride. Water can wash them around, but tiny critters are really important dispersers. Microarthropods like mites and springtails. Even bigger animals, birds, bats, maybe mammals that disturb the soil. There's a funny anecdote in the text about mites in the lab. You leave some cultures out, and pretty soon, mites track the spores all over your other clean plates.
Huh. Accidental contamination via mite taxi.
Exactly.
And speaking of weird interactions, tell us about Dictyostelium caveatum. That one sounds like something out of a sci-fi movie.
Oh, Decaveatum is fascinating. It's a predator. Found in a cave in Arkansas, its amoebae do something incredible. They actually invade the slugs, the pseudoplasmodium of other Dictyostelid species.
They get inside the slug.
Yeah, they slip in, and once inside, they start eating the host amoebae from within.
No way.
Yes, and the host slug just keeps moving, unaware it's got this internal parasite. It goes completely undetected until it's time for the host to form its sorocarp. But instead of the host species, suddenly Decaveatum pops out and makes its spores. The researchers must have been pretty surprised.
That's some sneaky cellular warfare. Okay, so bringing it back to the history, when did scientists first notice these things, and how did they become such a big deal in research?
Well, the very first one, Dictyostelium eukaryoids, was spotted way back in 1869 by Oscar Breyfeld. E.W. Olive wrote about them early on, too, 1902. But for decades, they were mostly seen as, kind of oddball curiosities, maybe a bit rare. The game really changed thanks to researchers like K.B. Raper and C. Tom, and then especially J.T. Bonner starting around the 1930s. Raper found Dictyostelium discoideum in 1935, and that species turned out to be common and relatively easy to work with in the lab. That really put them on the map.
And discoideum became the star, right? The model organism for studying how cells differentiate.
Absolutely. It went from being obscure to being the subject of hundreds of research papers every year. A lot of that research focuses on those signal molecules, like ACMP and DIF, that control how cells respond and change jobs. And understanding those signals in dictya, as researchers call it, has given us huge insights into cell communication and development in basically all multicellular life, including humans.
So their classification has shifted too. They were once lumped with another group.
Right. Initially, E.W. Olive put the Dictyostelids and another group called the Acytostelium together. They look superficially similar, maybe aggregating. But later, L.S. Olive recognized they were fundamentally different and separated them. It was a key taxonomic move.
How are they different?
Several ways. Dictyostelium usually have these thin, pointy pseudopods phalos. Acytostelids have more blunt, lobe-like ones, lobose. And the aggregation signal, the acrosyn. We know it's often CAMP in Dictyostelids. For acytostelids, the specific chemical signal is still, well, mostly unknown. Big difference.
And within the Dictyostelids themselves, how do scientists tell the main groups apart? You mentioned Polysphondylium earlier.
Yeah, the structure of the sorocarp, the fruiting body, is key. It's like their architectural signature. There are three main genera. Dictyostelium usually has stalks, sometimes branched, but the branches aren't arranged in neat circles or whorls. Then you have Polysphondylium. That's the one known for having branches that come off the main stalk in distinct whorls. Often looks quite elegant, sometimes violet-colored. And the third is Acytostelium. Its unique feature is that the stalk isn't made of cells at all. It's in a cellular tube that the amoebae secrete.
Different blueprints for building spore towers. And there's that story about Commononia, a lost genus.
Yes,
Yeah.
Nominoni. Described back in 1884, apparently had a really unique structure. But here's the thing, nobody's definitively found it since. For a long time, there was supposedly a reward offered for its rediscovery. I think the source mentions a case of good whiskey. It just highlights how elusive these tiny organisms can be.
A mycological mystery prize. Okay, just briefly on the genetics. Anything specific revealed there?
Well, studies looking at the genetics of those microcysts, the sexual stage, have shown some interesting things. For instance, sometimes not all the haploid cells produced after meiosis actually survive. Some genetic combinations might just not be viable.
And what about gene flow in the wild? The source mentioned some confusion there.
Right, that was an interesting puzzle with discoideum. Early studies using a DNA fingerprinting technique, RFLP analysis, suggested there was quite a bit of genetic mixing happening in natural populations. It looked like sex was common.
Okay.
But then, researchers tried directly crossing different strains in the lab and looking for offspring with mixed genes recombinant progeny, and they consistently failed to find them.
So the direct experiments contradicted the fingerprinting.
Exactly. The failure to get recombinants in the lab was seen as pretty strong evidence that maybe actual sexual reproduction and gene swapping isn't as frequent in the wild as the RFLP patterns first made it seem. It's a good reminder that interpreting genetic data needs care.
So after all this incredible biology, the social slugs, the self-sacrifice, the weird sex lives, the predatory behavior, what's the bottom line? Why should we, or maybe a college student studying biology, care about these slime molds?
Well, there are two big reasons, really. First, ecologically, they're important decomposers. They're part of that crucial cycle that breaks down dead stuff and recycles nutrients, especially by eating bacteria. They're tiny but mighty players in soil health. But arguably, their bigger impact is as a model system, studying how Dictyostelium amoebae communicate using CAMP, how they decide to become stalk or spore, how they cooperate. It teaches us fundamental principles about cell signaling, differentiation, and the evolution of multicellularity. These aren't just quirks of slime molds. These processes are echoed in our own bodies, in development, and how our cells talk to each other. So understanding Dicty helps us understand ourselves and life in general much better.
It really is an amazing journey, from individual amoeba to this complex cooperative slug, and through these intricate life cycles, a whole hidden world doing extraordinary things.
Absolutely. Yeah. And their value is a relatively simple system to study these incredibly complex universal biological questions. Cooperation, differentiation, signaling is just immense. They show us how much we can learn from even the seemingly simplest organisms.
It really makes you think, doesn't it? How studying these tiny, almost invisible creatures living in the soil can unlock such deep insights into how cells work together, how life builds complexity from simplicity, processes that are happening everywhere all the time. Well, thank you for joining us on this fascinating deep dive into the world of Dictyostelid cellular slime molds.
Hmm.